Sorry about that. My watch is running a little slow. It told me it was still 4:40. So, all right. So the plan for today is we're going to go over our brain lecture, and I wanted to also go over the leg muscle model. So I probably will start with that, and then we'll go over the brain lecture. The brain lecture is pretty long, so sometimes I'm not able to cover all of it in one session. But again, like we usually say in SI, the goal is not to go over like every single thing. It's kind of to get like an outline overview. So I will hopefully be able to go over all of the brain lecture today, just in case we can't. I'll pick up the rest of it Thursday morning, but hopefully we should be able to do all of it. Then on Thursday morning, we will go over the brain model and the spinal cord lecture. Okay, so today brain lecture, and then Thursday spinal cord lecture. Do you guys have any questions for me at all about lab, maybe about your lab packet or studying? Don't forget, you have your guide to muscles, right? And guide to muscles has all of your resources for the correct answers to the lab packet. If you haven't started labeling your muscles and doing the origin, insertion, action, innervation for them, you can find those answers in the guide to muscles. Again, if you're taking Bender's class, that would be in the study resources module. It's a document that kind of tells you how to study and what you need to know for the exam. Really short, it's just like 2 paragraphs right here. And then it also gives you would need to know this arm muscle list on the arm model. Again, you have your leg model, your arm model, right? So your arm muscle list, leg muscle list, torso muscle list, and then a muscle packet list. And then this muscle packet list is all of the answer key. And then you need to write those origin, insertion, action, innervation. And you also need to memorize, right, all of the stuff in that packet, and as well as all of your muscles. So for me personally, when I was in the class, we did not have access to muscles at home, just like you guys. Actually, I took all of these photos so that I would have access to the muscles, like to study them at home. Personally, for myself, that was not the best way to study them. So I actually went to the lab for extra lab time. When I took the class, Knutson was in lab Monday, Tuesday, Wednesday, and Thursday of every week. Now he's just there Tuesday, Thursdays. I believe Bender is there Mondays and Wednesdays, but you also have access to the tutoring center throughout the week, depending on what their hours are, and they have the muscles available as well. So the way I studied the muscles that was effective for me was going on campus. I really would tell you that probably studying the pictures isn't going to be super, super helpful to you. I would highly recommend going in person, kind of because you have to like lift off these little knobs and everything. It's not a bad way for us to review when we study them, but again, for you guys to learn them, I would recommend going to campus either for extra lab time or going to the tutoring center where you guys are at right now in this Zoom meeting, but it's their physical location. It's right next to your lab. It's like on the same story downstairs where your lab is. And again, they have muscles that you guys can use. The way that I studied the muscles when I studied them was the top part of a muscle and then the bottom part. So for example, I would study the upper part of the leg kind of until I could recite it, kind of get it down. And then I would do the lower part of the leg. The muscles are pretty easy to do because they have numbers. So on the top, for example, let's say you're studying you can take a piece of paper and you can, whoa, sorry, that's really close, but you can see it, 20 and 24. So you could take a piece of paper and you could write down, like, this is how I used to study. You kind of like, so I would write down, oh dear. Like I would write 20 and then I would write what I guessed that muscle was. So let's say I thought that this was rectus femoris, right? And then 24. These are not correct answers, but I'm just doing like, this is how I would do the work. And then what I would do from there is I would at the end go check my work. So I would pull up my key for the leg muscle list, number 20, rectus femoris. Oh, I got that one right. So then give yourself a check mark. Number 24, sartorius, not soleus. So then I would X that out and I would kind of do this exercise or this practice with the top part of the leg until I was getting most of them right. Does that make sense? So I obviously kind of understood it. And then I would move on to the lower part of the leg. Okay, so I would kind of quiz myself using the numbers. So these are a lot easier to study than the bones because of that, right? Because the bones are actually a lot more challenging because on the bones, there's no numbers. So you guys have to memorize all those little bumps versus on this exam, you have those numbers to practice with. So for labeling this picture, like I said, sorry, I have a little bit of a cold. This is rectus femoris, and then we have our sartorius, kind of like this muscle that goes sideways. Maybe you could think sideways, sartorius, sideways, going at an angle. And then we have, I'm going to rotate it to see this one. This muscle over here is going to be your vastus lateralis, and we'll rotate it to see the side. Those are kind of the best three we can see right here. And like I said, we'll rotate it. So again, this one right here would be your sartorius, but seen kind of from the side angle. And then we have our sorry, that's not, there we go. Let's get it out a little bit. This muscle here is the vastus medialis, right here. And then we have this flat one right here that goes up and down. And I remember this is called gracilis. And I remember this because grass is straight. So you know how like a blade of grass is like runs straight up and down. So that's how I remember gracilis, because it runs straight up and down the center of your leg. So that's how I remember that one. And then we have adductor longus towards the front, and then adductor magnus towards the back of gracilis. So you could remember that as L comes in the alphabet before M. So adductor longus, and then adductor magnus. Another way you could remember it is magnus is on the same side as the gluteus maximus, which is the butt, right? So the magnus is on the gluteus maximus side, right? So again, longus is in the front of gracilis, magnus is in the back. Does that make sense? They're like the two that go on either side. Okay. All right, I'll see you guys out there.
Recording in progress.
All right, and then on the back of the leg, this one has the gluteus maximus removed. So let me try to find you guys a picture with it. There. So here's the gluteus maximus in place, right? The next picture, they removed it so we can see the muscles underneath. So I don't need to label gluteus maximus, I don't think, because that one's kind of straightforward. This is gluteus medius. We have piriformis. I believe this is, let me check. Piriformis, gemellus superior, right here. Obturator internus and gemellus inferior. You probably likely started here. Gemellus superior, obturator internus. You could think of like it's internal or the one like wedged in between. Obturator internus. And then the last one, gemellus inferior. So I think of them as like the little gems, like they're really tiny, like gemstones. So gemellus superior is the one on the top, inferior is the one on the bottom, and obturator internus, it's like it's wedged internal in between the two. Or you could think in between internus, obturator internus. And then our last one here is quadratus femoris. And then, like I already told you, this guy right here is our vastus lateralis. And then we also have the iliotibial tract. Iliotibial tract. Okay. All right, any questions on this guy right here? As you guys already know, if you guys leave early, you need to put that in the chat, okay? All right, so let's do a few more views of the leg. Sorry, my voice is kind of weird. And then we'll move on to the brain lecture, okay? Let's see, so this is our leg with the rectus femoris removed, right? I had shown you guys the view with the rectus femoris in place. So underneath the rectus femoris is the vastus intermedius. Okay, so what you have the vastus medialis on the inside. The vastus intermedius is in the middle, like intermediate. And then out here is vastus lateralis. So it's kind of easy to remember. They go together, right? Medialis, medial, intermedius in the middle, and lateralis on the side. We have our adductor longus, our iliopsoas. You'll see on the models, there's a way to remember this. So there's this little muscle right here. Do you guys see this one where I put the P? So it's IPA is how you remember it. But you don't have pectineus on this model that you'll be tested on. So that's why I just put a little P. But when we look at the torsos, you'll see the P, the pectineus bigger. But you can remember this still with like IPA, right? That's how you can kind of remember what this one is, iliopsoas. Okay, it's the I, iliopsoas. Good, and I already showed you gracilis is this one here. So that should be most of the stuff for the upper leg. I'll rotate it just to make sure I didn't miss anything. And then, oh, I need to do the two on the back. And then we'll go to the bottom of the leg. Here's another view of the front. This is the tensor fascia latae. Most people really like this one because it's it has the word latae in it. So tensor fascia latae. So that is the one right here above the iliotibial tract and vastus lateralis. Right, so again, this would be vastus lateralis right here. This is your rectus femoris right here. vastus medialis. Vastus intermedius is underneath the rectus femoris. And then the iliotibial tract is this white part here that connects that tensor fascia latae to the knee. Okay. And then this is the tibialis anterior. We're going to get down to the lower leg in just a second. Tibialis anterior is that muscle. Okay. So you could use these pictures to study at home as like a way to like quiz yourself. But like I said, I do think that the best way to study these would be going in person and seeing the legs in person. All right, so our last view of the legs is our two large structures here, and then we should have something underneath we need to know as well. Maybe I already did the underneath part. So there's our two big muscles. The way I remember it is there's these two that come in a pair, right? And then there's the other two. So we have 30, 31, and 30. So 31 is our long head of the biceps femoris, long head of the biceps femoris. And then out here to the side, these two that come in the pair are semitendinosus and semimembranosus. The way I remember it is tendon is like more superficial, like closer to your skin, and then a membrane would be like smaller and thinner. So if you look, semimembranosus is this little guy down here that's closer to the actual leg versus semitendinosus is more superficial, right? And then this one back here, we've already gone over it, is adductor magnus. There's also a short head of the biceps femoris, and then we can only see that when we take the muscle out, okay? So semitendinosus and semimembranosus, I remember that because they're always like in a pair like that, where you can see the membranosus right underneath. All right, let's look underneath really quickly. So this shows us that short head of the biceps. What do I feel like? Is it called biceps femoris? Why is that sounding weird? It is sounding weird. It is called biceps femoris, which sounds like it should be on your arm, but your arm is called biceps brachii, right? So this is short head of the biceps femoris. The long head of the biceps femoris was this one that's laying down on the table. Okay. All right, any questions there? So that wraps up the upper leg, and then we'll do the lower leg. So looking at the lower leg, usually we start at the front. So this one's kind of hard to see because it's a little bit blurry. We'll see if we have a better view up front. This one's a little better here. All right, so this is our tibialis anterior right here. And then this is a little bit, there we go, that's a little better, it's a little closer. So this guy right here, 34, it's flexing the toes, right? Because if you think about your hand, right? So the palm of your hand is like the sole of your feet. So this would be the extensor side of your feet, right? So sometimes on the test, I used to have to like do that. Just think of the palms of your hands are like the soles of your feet. So flexors are going to be on your calf side, extensors are on this side. So this muscle right here is going to be an extensor, right? It's the extensor digitorum longus. You can see that it's going to the digits, the toes, and then it's long, extensor digitorum longus. See if we can get it to extend that right here. All right, and then we have an extensor digitorum brevis as well, which should be these little guys here. I can't see a number right now, but when we rotate it, we should be able to see. So these little muscles here that are kind of like extending the toes should be extensor digitorum brevis, but I'll label it when we get over to the other side, okay? And then we have our extensor hallucis, our extensor hallucis muscles, which should be 35. No, that's not the right label. Let me zoom in on it and see what number it is. 44. So 44 is our extensor hallucis longus. Hallucis stands for big toe. So remember we had pollicis for thumb. So now it's hallucis. So extensor hallucis longus. So I'll draw it over to it because it's not on the right spot. Right here. So this little muscle here going out to the big toe is our extensor hallucis longus. 45 could represent the extensor hallucis, right? Because like I told you guys, 99% of the time on these muscles, you're going to always have a brevis for a longus. So that 45 that you see right here on the big toe itself is your extensor hallucis brevis. Extensor hallucis brevis, right? It's extending the big toe. There's the longus on the actual calf, and then we have the brevis right here. And then remember how on the back of your hand, what was the muscle called on the back of your hand that goes in between all of your fingers? It's called the dorsal interosseous, right? So that's the dorsal interosseous. It's the muscle that goes in between the toes, okay? This other muscle over here is going to be extensor digitorum brevis, but like I already said, we'll see that when we turn. Dorsal interosseous would run up and down the toes versus the extensor digitorum brevis is going to be like these ones right here that are kind of running sideways. Okay. And then I'll label these two guys when we rotate it because it's kind of hard to see them well right now. So again, we're on the extensor side. And if you guys have questions at any point, please write those in the chat, or you can always unmute yourselves and ask. Okay, so again, like I had said, these should be, can't even see the number. I think it says 36. 46. Just make sure. 46 extensor digitorum brevis. So like I said, the longest is on the front, and this is the extensor. This all caps is really annoying. Extensor digitorum brevis. Okay. So those are those ones that kind of go sideways. This is going to be our abductor digiti minimi. Abductor digiti minimi. Remember that the digiti minimi just means pinky. So in this case, it's referring to your pinky toe. There's not a longus and a brevis. It's just abductor digiti minimi. These are the peroneus or fibularis longus and peroneus or fibularis brevis. So this is the longest that I just showed you here. And this one on the side, peroneus or fibularis brevis. If any of you guys were paying attention, right, when you guys were studying your bones, you would remember that your fibula had a tendon, a groove for the tendons of peroneus longus and peroneus brevis. Do you remember that? So that's right here. If you look, we're on the bone, we're on the fibula, right? And there's our groove. Does that make sense? So it kind of can help you remember where they would be located. The longest is the one that looks long, and the brevis is the one that looks short at the bottom. Okay. This large muscle at the back is our gastric memmeus. And then there is the soleus muscle, which is the one right underneath, soleus. That's this kind of like second crease. And then there's one bonus term you guys need to know for this exam that's not on your list. I think it's just we haven't gotten around to putting it on your list. So Achilles tendon. So your Achilles tendon is right here. It is not on your list. And you do need to know that for the exam. All right. Okay, so I'm gonna keep moving. Go to the back of the leg and the foot, and then we'll work on the brain flexure. So again, we did the extensor side, the side of the leg, and now we're going to be doing the flexor side or the back of the leg. And the leg is a lot easier than the arm. So if you're struggling on the arm and you started with the arm, that's fine. Just know that the leg will feel a lot easier when you get to the leg. So that's again the gastrocnemius, the soleus, and let's change our color here. Achilles tendon. Okay. There's a popliteus muscle in here. I don't think we can see it from this view. I have to move aside this thing before we can see it. All right, let's move that and look underneath right here. So we have the popliteus muscle, and then the tibialis posterior and the flexor hallucis longus. So let me make sure I have my numbers right. 40. So tibialis posterior is this one right here. This one. Tibialis posterior. And then our flexor hallucis longus, it kind of goes in the direction of that big toe, right? This muscle here. So that kind of makes sense. Flexor hallucis longus. And we should see the brevis on the actual foot itself. And then the last muscle that's a part of these three is the popliteus. So when you guys peel off, like on your test, when you take this off right here and you pull it off, there are three muscles to know on the back. The way I remember this, well, I guess that actually doesn't make sense because that wouldn't, wait. Oh, there's one more, isn't there? There's a flexor digitorum. 43, let me make sure I have this. 43, flexor hallucis longus, 42, flexor digitorum. Once we rotate right here, you'll see this guy, and it's the flexor digitorum longus. Flexor digitorum longus, this one. It's so hard to do these with a computer because it's not as fast, but we're almost there. And then like I said, I'll go on to the brain next. Okay, so those are the ones that we see on the back. And then we'll rotate it one more time and then look at the foot and then we'll be done, okay? So this is just kind of like a good little recap for you guys. If you have been studying them and you kind of wanted to do like a little review of them. Let me see if I can get my side view. I think it shows it right here. So those are the two. This should be your flexor digitorum longus right here. And I already kind of labeled it on the previous one, so I'll just let you guys find that. All right, so our last view of the foot, and then just to kind of give a little heads up, on Thursday, I will go over the torsos. Okay, so today we're just doing leg, and then on Thursday, I'll go over some of those torsos so you guys can have all the muscles gone over at some point here in SI. All right, so we're on the last part, we're on the bottom of the foot, okay? So we have our flexor hallucis brevis, right? We already said there was the abductor hallucis, and now this little guy right here is your flexor hallucis brevis. And then we have the muscles that go in between the foot on the bottom, and those are the lumbricals, just like on the palm of your hand. You had the lumbricals, and I told you it was kind of like remembering to throw that brick at someone. And then 48 is one of your only one that you need to know that is brevis that doesn't have a longus. So this is the flexor digiti minimi brevis right here. Flexor digiti minimi brevis. And like I tell you, there's always a longus and a brevis. Not in this case. This time we only have a brevis, okay? And then very last, we have our flexor digitorum brevis. It should be. Let me go make sure though. I don't want to say the wrong thing. Flexor digitorum brevis. So again, flexor digitorum longus was located on the back of the leg. Here's the brevis. You can remember because it's flexing those digits, and it's the short version because it's on the foot itself. Okay. And then that is everything for the leg. And then we'll go and we'll work on the brain next. Any questions at all on that? Okay. I want to remind you guys that once again, you have access to the videos of the previous SI leader going over the models, if you guys want to take advantage of those. And also, there are a lot of the different practice questions for you guys, just like the other on the last exam. So if you want to access to practice questions, those are there for you as well. Okay. So let's do our usual kind of outline here. So brain and cranial nerve. So that's the title of the lecture, is the brain and the cranial nerves. So I try to make this lecture as organized as possible. It does require a little bit of reorganization, but I try to kind of make it as like simple and understandable as we can. So there are 4 regions to the brain. So the lecture kind of is divided up into like some basics, like some about information about the brain, but for the most part, we spend our time talking about the 4 divisions of the brain. So we have our cerebrum, our diencephalon, the brainstem, and the cerebellum. So those are the 4 regions, and then we get into details below those. So for the cerebrum, it has convolutions on it. So I'm kind of I'm sort of reorganizing the lecture a little bit. So if you guys have your notes and you're following along, you might see me kind of recategorizing, but in your notes, it says convolutions in the cerebrum, and they're there to increase surface area. And then we have basically 3 types, right? We have our gyri. Gyri is multiple, gyrus would be singular. And I'm going to put an arrow up that represents the fact that there are rise in the convolution. And then there's the sulci or the sulcus, representing that they are a depression or a down in the convolution, and then a fissure, and I'll put 2 arrows down. They're a very deep fall. So fissures actually divide the cerebrum into lobes. So the cerebrum has 5 lobes. So there are 5 lobes, and we'll talk about those. And we may not have space on this outline to like go into detail about each thing. So I'll just write those here. So we have frontal, parietal, temporal, occipital, and then the insula. Okay. So one of the basics about the brain is that it has the directional terms rostral and caudal. I'm kind of just throwing these up here because, like I said, there's a few random things that don't fit into the 4 divisions. Rostral, you can think of like nostril. Rostral refers to like anterior. And then caudal refers to the tail. So this would be posterior. So this allows directional terms for the brain, no matter where someone is positioned. That is always true for those areas, okay? Then we'll talk about gray matter versus white matter. This is kind of like another little area or topic we'll talk about, gray versus white matter. And then we'll talk about meninges. And we'll do that on a new page just because they don't really fit super well right here. And then again with cerebrum, we'll kind of go into more detail about the 5 lobes and just kind of trying to give you an outline of the different ones. Then for diencephalon, we have your epithalamus, thalamus, and then hypothalamus. And I'll come back to this page and add more detail. Brainstem, you have your midbrain, pons, and medulla oblongata. And then lastly, the cerebellum, which does not have different parts. Okay, so the midbrain, the brainstem and the diencephalon both have 3 parts. The cerebrum has 5 parts. They're called lobes, 5 lobes. The brain itself has 4 regions: cerebrum, diencephalon, brainstem, cerebellum. Cerebrum has 5 lobes, diencephalon has 3 parts, brainstem has 3 parts, and cerebellum does not have parts. We just talk about the cerebellum function as a whole. Okay, so we'll come back and we'll kind of fill that in. So let's kind of talk about the brain in general, and then we'll go back and we'll fill our outline in and we'll add some more detail, okay? So overall, there's some terms you need to know with the CNS. I'll see if I can make a little outline out of this. So there are basic terms that are really important that you guys know for the test. So in the central nervous system, we have the word tract that references a myelinated axon bundle. So we get all these different little axons together in the central nervous system, which remember is the brain and spinal cord, and we call that a tract. So if there's a lot of axons together in the brain and the spinal cord, it's called a tract. But if we have the actual cell bodies of those neurons, because remember we have the little neuron, right? And it has the cell body. So let's say we have a lot of those little cell bodies together. That is called a nuclei. So clusters of cell bodies, nuclei. When we get multiple nuclei together, and it's like a lot of neurons that share the same function, bunches of nuclei that share the same function, that's called a center. So for example, you might have a center for vision. There's a center in your brain for vision. You might have a center in your brain for smell, vision, and there are more, right, etc. And then when multiple tracts get together, we call that a funiculus. A funiculus is a large tract. So on the test, your questions might ask you, which of these is the center for hearing? So you need to know what the word center means in order to answer the question. Does that make sense? So you guys just need to make sure you go over these different terms for the exam so that you're actually prepared to answer it in the form as part of another question. Then we have the peripheral nervous system. Peripheral nervous system. Remember, this is everything that comes off the brain and spinal cord. So the peripheral nervous system has the same thing as a tract, a myelinated axon bundle, but we call it a nerve. So on the test, you need to know that a nerve is only part of the peripheral nervous system. When multiple nerves get together, that's called a nerve plexus. So kind of like a funiculus, a nerve plexus is a network of nerves. And then finally, we have our ganglion. And a ganglion is a cluster of cell bodies. You may not have, like Knudsen didn't explain this. There's a table in your book, and I made this chart when I was taking the class in order to like interpret the table in the book. Does that make sense? So you probably won't see this in your notes, but it is something that's kind of beneficial. It's like a simplified version to make it easier to remember for your exam. So that's one little part of kind of the brain and how we talk about the brain. Then we have the protective covering around the brain, the meninges. This is the same as the sac around the brain. Remember, we have like the sac around the lungs, the sac around the heart. This is the sac around the brain. So it's 3 different connective tissue layers. We have our outside, which is called the dura mater. You could think of it, it kind of sounds like it's durable, right? And you would want your outside layer to be durable. So this sticks against bone. And remember, this is for the central nervous system. So in the brain, it's the cranium, that's the bone. And in the vertebrae, it would be the vertebral canal. Remember when you guys learned about vertebral canal on the bones? So that would be for the vertebrae. Well, I guess it would be for the vertebral column, would be the spinal cord, right? Sorry, I was looking for the word spinal cord and it wasn't coming out. Spinal cord. And then we have our arachnoid mater. Everything or myelinated. Someone asked myelinated. Arachnoid mater is the next layer. Arachnoid means spider because it has spider-like, it looks kind of like a spider. And in the, I'll draw this in red because it's kind of important. So the subarachnoid space has, I'll draw that in yellow, CSF. Everybody remember CSF? Cerebrospinal fluid. And I'm going to do a little thing here. Where is cerebrospinal fluid made? It's made by the astrocytes and the ependymal cells, right, that line the ventricles of the brain. So the ventricles of the brain store CSF. So store, where is CSF stored? In the ventricles. Where does CSF circulate in order to pass to all the different neurons? It circulates in the, I'll write sub-A space, the subarachnoid space. And then how is CSF reabsorbed back into the circulation in order to be filtered by the kidneys, right? So you make this purified blood. We know CSF is purified blood, right? It's blood without any red blood cells, white blood cells. It just has nutrients. So only nutrients. So where is that created? Astrocytes and ependymal cells lining the ventricles of the brain create that blood-brain barrier, right? BBB, blood-brain barrier, that makes it. It's stored in the ventricles of the brain. It circulates in the subarachnoid space and it's reabsorbed by structures called arachnoid villi. So arachnoid villi take up that old used CSF and they return that used CSF to the blood. Arachnoid villi stick out into sinuses. So I'm going to put sinuses. Sinuses in the brain. These are not like your nasal sinuses. So if we were kind of, I'll try to draw like a little representation. So here's my brain and spinal cord. Here is, we'll draw a little like, this is my subarachnoid space. Okay, subarachnoid space. In it, you have these like little, let me see if I can, this thing is not cooperating with me. This, let's say that this is a little sinus. Okay, here's a sinus. They're like little collection points, sinus. The sinus has little arachnoid villi. We'll kind of do them like this, like little fingers. And those little arachnoid villi pick up the old used CSF, kind of like your lungs put carbon dioxide, oxygen back into your blood, right? From carbon dioxide gas exchange, arachnoid villi take away old CSF and return it to the blood. And then the ventricles of the brain kind of look like little, there's actually 4. So there's lateral, there's 1 lateral on each side. So that's 2. And then there's a 3rd and a 4th. And yes, you need to know that there's a 3rd and a 4th and 2 lateral for your test. So these are your ventricles. Okay, ventricles, there are 2 lateral, a 3rd and a 4th ventricle. Okay, and connecting the 3rd ventricle to the 4th ventricle is what's called the cerebral aqueduct. So I'm going to draw like a little tube. Cerebral aqueduct. And this connects the 3rd to the 4th ventricle. So all of the ventricles are kind of connected. The 3rd ventricle is located in the diencephalon. Yes, you would want to know locations for the test. 4th ventricle is located between the pons and the cerebellum. Cerebral aqueduct connects the 3rd to the 4th ventricle. And lastly, I'll draw it also in blue. The 2 lateral ventricles are separated by the septum pellucidum. Okay, so this separates the lateral ventricles. Okay. All right, so again, ventricles store CSF. It circulates in that subarachnoid space, reabsorbed in the sinuses by the arachnoid villi. And then we have our 2 lateral separated by septum pellucidum, 3rd and 4th connected by cerebral aqueduct. And then our very, very last layer, number 3, I know that was a lot kind of on arachnoid mater, but the very last layer is our pia mater. And the pia mater sticks to the brain and spinal cord. So it's like a very thin membrane that is sticking to that brain and spinal cord. All right, so those are the cranial meninges. Very important there, and that's kind of like a pretty good summary right there on that page of what you would need to know. Sorry, I just realized you guys were, I think you got it. Ganglion, yeah, starts with a G, ganglion. Sorry, I know that was late. I saw you guys answering each other. And we'll talk more about a ganglion, but just know it's a cluster of cell bodies in the PNS versus nuclei, cluster of cell bodies in the CNS, right? Tract, myelinated axon bundle, CNS, nerve, myelinated axon bundle, PNS, right? So make sure you guys get that. This thing's not right. Okay. All right. Any questions there? We'll talk about gray and white matter, and then we'll get into the parts of the brain. Before we leave this page, CSF does have some different functions. I'm going to kind of do this so that I can write them right here because I don't really want to leave the CSF page. So this is pretty simple. It's another reason I don't really want to leave this. So functions of CSF It provides buoyancy, which means it helps your brain like float and not get damaged. It provides your brain protection, right? Again, you're floating in liquid so that when you bonk your head right on the car door when you're coming inside, you don't have a head injury and damage all your neurons. It gives you that liquid. And then it provides stability, environmental stability, which basically means it helps to pull away waste products, right? And to provide nutrients, which we've already talked about. If you have an infection of your meninges, it's called meningitis. These aren't super likely to be tested. Hydrocephalus is in babies if this cerebral aqueduct becomes blocked, then it's called hydrocephalus. So this can cause a baby's head to be very big. As basically the fluid, the CSF, cannot leave the brain and the cerebral aqueduct being blocked. And we call that hydrocephalus, which means water on the brain. There's too much water in the brain because that cerebral aqueduct is blocked in babies. It's really probably not going to be on your test, but I don't want to say that with 100% certainty. It's not too complicated to know. So just know hydrocephalus is if that cerebral aqueduct becomes blocked for some reason, okay? All right, any questions there? All right, so we're gonna keep going. We're gonna talk really quickly about gray versus white matter, and then we'll get going into the actual other areas of the brain. Yes, we're gonna come back and fill this in just a minute. You said you wanted to see this. All right, I'll come back to it though, okay? Because we're not done filling it in. So lastly, for the brain, and this also applies to the spinal cord. In both, right, in your central nervous system, we have gray matter. This is a G. Now I realize my C's and G's look like. And then let's do blue to represent white. So we have gray matter and we have white matter. I actually prefer, I wish I could do yellow. But just leave it there. I'd rather do. Okay. So for your gray matter and your white matter, the easiest way to do this is to memorize the definition of white matter. So white matter represents myelinated axons. So if we draw a little neuron, I draw a little neuron here. Here's its axon, right? And its telodendria synaptic knob. If it has myelin on it, that belongs to white matter, okay? Now, if it is unmyelinated, which can happen, some neurons do not have myelin. Most neurons have myelin. We already talked about the functions of myelin, but some neurons don't. So everything besides for that myelinated axon will be present in the gray matter. So we'll draw like a big X through that part, and then we'll say all the rest. So what is left over? Unmyelinated axons, right? And then let's look at all the other parts of the neuron. Cell bodies, right? Dendrites, telodendria. So just think of your anatomy of the neuron. Synaptic knob. All cell bodies, right, that exist in the CNS. So CNS cell bodies. So you kind of are still getting into all this, so I'll just remind you of what those two are. So there's the motor neuron and the interneuron. The sensory cell bodies are located in the dorsal root ganglion of the spinal cord. And they are unipolar cell bodies. I'm just telling you again so that you can know. This means that they are PNS, they're in the peripheral nervous system. They are not gray matter. So just make sure you realize I'm not saying that this sentence is representing gray matter. I'm just showing you motor neuron cell bodies, interneuron cell bodies, those would be in the CNS. But our sensory cell bodies would not because they're in the dorsal root ganglion. Remember from our previous page, what is a ganglion? It is a cluster of cell bodies in the PNS. So let's go back over here. The dorsal root ganglion represents a cluster of unipolar cell bodies in the PNS, right? All right, gray matter represents the cortex of the brain. This means it's like the outer layer of the brain. And then we have our cerebral nuclei, so it's the cortex or the outer layer, and the cerebral nuclei, which are deep regions of gray matter, deep regions. And then our white matter is only the myelinated axons. And then lastly, the corpus callosum is your largest tract in the brain. So remember, this would be nerves in the PNS, tracts in the CNS, and the corpus callosum is the largest tract in the brain. So the white matter is to help facilitate communication between the gray matter. So for example, here's my brain. I have my cortex, which is that outer gray matter. And then I have these deep clusters of gray matter that are called cerebral nuclei. They're kind of around the brainstem. So the white matter is going to be connecting all of the cortex to the cerebral nuclei, right? Because if you visualize what white matter is, it's just, here's my cell bodies and maybe my telodendria, or maybe this is a cell body, this is a telodendria, right? And this is just the myelinated portion. So you need to think of that when you're thinking about what is white matter. It's the myelinated portion. What is gray matter? It's everything that it's linking to back and forth across the brain. That makes sense? All right. So we're going to talk about the cerebrum. Okay, so I'll try to fit what I can on this outline because I think it's kind of nice to see everything we can on the outline. And then when I run out of room, we'll go to the other part. So the cerebrum, the function of the cerebrum provides us the ability to reason. Yes, let me go back. So again, function of our cerebrum provides us the ability to reason. So that cerebrum has the outer cortex. We just talked about this, right? Outer cortex. Inner white matter, those myelinated axons, matter are just the diencephalon, the brainstem, and there's white matter and gray matter in the cerebellum. Yes, the cerebrum has the majority of gray matter and white matter because the cerebrum is the biggest part of your brain. It's the big thing you see when you look at somebody's brain, you see cerebrum. But there is white matter and gray matter in the other section. So that's why we don't just get that's for this part. And there are white matter and gray matter in the spinal cord as well, which we'll get to on Thursday. The cerebrum is divided in halves. These are called the cerebral hemispheres. So these are the halves of the cerebrum. Cerebral hemispheres, it's divided in two halves. I'm just going to kind of use a little bit of the bottom of the screen here. So the cerebral hemispheres are divided in two halves. What separates the halves of the cerebrum is the longitudinal fissure. And the corpus callosum helps connect those two halves. Helps connect the two halves. So again, cerebral hemispheres, I have a left and a right. They are connected, they are separated by a longitudinal fissure, and the corpus callosum helps connect them. So because you have two halves, you've heard people say you have a left side of your brain and a right side of your brain. Most of the time, this Apple Pencil is like, most of the time your control is contralateral. So 95% of the time, this is like 95% of the time, the opposite side of your brain controls that side. So if I move my right hand, my left side of my brain told me to do it. Does that make sense? So I'm going to put like left. I don't know what is. Okay, I've got something going on my screen. Left to right. Then there's ipsilateral. Ipsilateral is 5% of the time, and it's same side. The same side controls the same side. Okay. Okay, so the frontal lobe is our first lobe of the cerebrum. So that's kind of general stuff about the cerebrum, and now we go into our lobes. Frontal lobe, primary function is motor. Parietal lobe, primary function is sensory, except smell. So motor, frontal controls what you do. Parietal lobe senses things except smell. Temporal lobe is for hearing and smell. Occipital lobe is for vision. And then the insula is for taste. That is like the outline format of it. I'm just going to make a separate page and we'll go into a tiny bit more detail about what you need to know there. But we'll keep going with diencephalon in just a second, okay? So that was cerebrum overview, the 5 lobes overview, and then we'll talk a little more about a few terms you need to know for the test for the lobes. So when we're looking at our 5 lobes, first lobe is frontal. Main thing you remember is motor. The other key term is that there is the precentral gyrus located behind the central sulcus, which is what divides the frontal lobe from the parietal lobe. So on the cerebrum, we have our frontal lobe here. Then you basically have it splits into your parietal lobe. If you remember on the skull, you guys remember learning about these on the skull, right? And then you have your occipital in the back. So what divides the frontal and the parietal lobe is this little line right here. This is called the central sulcus. Remember, sulcus is the down part of a convolution, so it's like dividing them. So precentral gyrus is going to be the raised portion in front of or anterior to the central sulcus. So the raised portion, why? Because that's what a gyrus is. So the raised portion of the convolution anterior to the central sulcus. So that's the frontal lobe. The main function of the precentral gyrus is voluntary skeletal muscle activity. So it's just important that you know for the test what it does, that it controls voluntary skeletal muscle activity to specific locations. So like if you need to move your tongue, there's a little section on the precentral gyrus that moves your tongue. You would also need to know for your test that it's also called the primary motor cortex. Primary motor cortex is also called the precentral gyrus. The next lobe, let's do a new color. Let's do blue. Next lobe is our parietal lobe. Again, these are lobes of the cerebrum. And we said it was for general sensory, right? Except the sense of smell. So parietal lobe also has a little gyrus that comes by the central sulcus, but we call this the post-central gyrus because it would be posterior in the brain. That term is rostral. So it's rostral to the central sulcus. Okay, central sulcus being that little divot that divides the frontal from parietal. It is also called the primary somatosensory, right? Sensing things from the body, primary somatosensory cortex. And like I already said, this is anything you sense except smell. So if you eat something and it feels very rough on your lips, that would be sensing something from your lips, telling your brain it's on that little section of the primary somatosensory cortex. So like my lips are right here, and then moving my lips is over here. So when I move my lips, it's on that primary motor cortex. When I sense something from my lips, it's on the primary somatosensory cortex. The other name is postcentral gyrus versus primary motor. The other name is precentral gyrus. Then you have your Broca's area, which is located on that motor portion. So this is the Broca's area. I'll try to kind of draw a little box. It's not a part of that primary motor cortex. It's a part of the frontal lobe. So we'll put frontal lobe. The Broca's area controls speaking and writing. The way I remember this, if any of you speak Spanish, I speak Portuguese. So the word boca, right, means mouth. So I remember it like mouth, like you're speaking using your mouth. So Broca's area controls speaking, writing, like to actually physically do it. You can also remember that it's located in the frontal lobe because it's an action, right? We physically decide to speak. We decide to write. It's an action. All right, our next lobe is the temporal lobe. Let's do green on that one. This one's pretty simple. It's just for hearing and smell. So again, smell does not go through the parietal lobe. Smell goes to the temporal lobe, to the primary olfactory cortex, okay, to the primary olfactory cortex, and it is the only sense that does not go through that post-central gyrus or the thalamus. So the thalamus on this outline, thalamus, is a switching station for sensory information, except smell. So the thalamus like redirects information, telling it where to go based off of that primary motor cortex or that primary somatosensory cortex. So again, the thalamus would kind of redirect to the primary motor cortex or the primary somatosensory cortex located on the frontal and parietal lobes, but not for smell, right? So the primary olfactory cortex, we have smell being the only sense that does not go through the postcentral gyrus or the thalamus. Another little kind of bonus term, I'll write it the same way I wrote Broca's. Wernicke's area. This is located in the parietal and temporal lobes, which makes sense because the parietal and temporal lobes are just for sensing. So this is how you understand what is written and spoken. So if you think about it, that's sensory, right? I hear you guys say something and I understand what you're saying. So for example, somebody who has a stroke or a brain injury might understand everything somebody's saying to them. The Wernicke's area is working great, but they can't speak. They can't say anything back, Broca's area. Or somebody might speak and be speaking just fine, but their Wernicke's area is damaged because they can't hear that they're saying the wrong thing. So this would be someone who's like talking to you and telling you a story, right? But none of the words make sense. You know, they're saying like cat, like it doesn't, nothing, cat ball jumped, elephant outside. And then you look at them and you're like, that didn't make sense. And they're like, cat ball, they say it like more forcefully because their Wernicke's area does not comprehend that what they're speaking doesn't make sense. Does that make sense? So you need to have both functioning to properly speak. Wernicke's, how you hear it and understand it. Broca's, how you actually speak it, the action of speaking. So those two are important to know. So we had our frontal lobe, our parietal lobe, our temporal lobe, and then we have our occipital lobe. Occipital. This processes and stores visual information. So it's very simple. It's just what you see, your visual, what you see. And then the very last lobe is also very simple. It's called the insula. And this is just for taste. And it also helps connects other lobes. So taste, and it connects other lobes. So that's the insula. And then our very last kind of like bonus term is the gnostic area. So like the Broca's area and the Wernicke's area, we have our gnostic area. You don't need to worry about its location, but I mean, we can just write it. It's located crossing the parietal, temporal, and occipital lobes. That's why we talk about it here. And its function is for knowledge and spirit. Kind of like that sense that there's another world out there, right? How we feel there's more beyond just this life. So that's the gnostic area, the knowledge or spirit located crossing over parietal, temporal, and occipital lobes. In your PowerPoint slides, you'll probably see there's quite a few association areas. You really don't need to worry about the association areas for your exam. Knudsen would have directed you to not worry about those, and I don't believe Bender has those on his slides. So again, for association areas, you don't need to worry about that. All right, any questions there? So those are the 5 lobes of the cerebrum, right? So we just covered the cerebrum of the brain. Okay. So going back to our gray and our white matter, I'm not entirely sure why this is kind of, some of the notes are set up a little strange, but in the same section of our gray and our white matter, we have what's called the tracts, the central white matter. So one of the example here, I'm going to put it right here, projection tracts. These help connect the brain. You're not very likely to see the tracts appear on your exam, so I don't want to spend too much time, but projection tracts basically look like this. They're kind of like these little connection points. You also have some different ones you'll see in your PowerPoint slide. You have commissural. They all represent white matter. They're just showing you that sometimes they cross at different points in the brain. Now this, you will see this appear on your test. So these are the cerebral nuclei. Like I already told you, those are the deep regions of gray matter, right? I kind of drew them over here, cerebral nuclei. They're kind of located deep. These are not found right on that cortex. They're deep and around like basically the brainstem. And you need to know some different examples. So you need to know amygdaloid body plays a role in anger or fear. The caudate nucleus is for the pattern and rhythm of walking. And then the claustrum is for subconscious visual information. These are also involved in the limbic system. I'm gonna go try to find the limbic system ones real quick. So I'm gonna just put on the limbic system one so that it's a little less of stuff being everywhere. So amygdaloid body, anger, fear, claustrum, subconscious visual. Then we have the limbic system ones, which are overlap, but with a few new ones. So I'll write them like this. So number 5, we have our hippocampus. Hippocampus stores long-term memory. Oh no, I shouldn't have done red. I have red for white matter. Let's do blue. Hippocampus is just part of the hippocampus, so there's no real like function to it. And then I need room for 2 more, so let's just erase this because I want to put them all in one spot because they're all cerebral nuclei. It's just that the blue ones also specialize in like being part of the limbic system. So for the limbic system, it's the amygdaloid body, the hippocampus, the parahippocampus, and then these last 2. So I'm going to do it like this because this will make it less confusing. 2, 3, 4, and then our claustrum. Well, we'll do claustrum. Claustrum is 4, and then 5. I'll try to explain this a little more clear. I don't want to confuse you. Cingulate gyrus is the last one, and this receives input from other limbic parts. Okay, so all of them are cerebral nuclei. The 4 examples of cerebral nuclei that were given to you are already given here in black. Does that make sense? Amygdaloid body, lentiform nucleus, caudate nucleus, and claustrum. In addition to those, there are 3 more. Amygdaloid body and claustrum are part of the limbic system. The lentiform nucleus and caudate nucleus are not a part of the limbic system. This should make sense because the limbic system, as you guys should remember if you watch the lecture, is for emotions. So unconscious muscle control is not emotional, right? No emotions there. And pattern or rhythm of walking is not emotions. So if you think about it, anger and fear, amygdaloid body is part of the limbic system because it's an emotion. Claustrum, visual information, subconscious, we can have emotions there. Long-term memory, we would have emotions. And the parahippocampus helps with that, so that's going to be involved. And then lastly, cingulate gyrus because it receives input from other limbic parts. So hopefully I didn't confuse you. The 4 main ones are cerebral nuclei, and then we add on a few more for the limbic system. Okay, you'll probably get a few questions on the test on those. And it can just be as simple definitions as what I just gave you. All right, so let's kind of go back to our outline and we'll talk about our diencephalon now. So kind of make it a little, things stand out a little bit better. So in the diencephalon, the main thing we have going on is hormones. So the diencephalon main function is hormones. We know that that's the endocrine. The epithalamus controls our sleep pattern, and this is via melatonin. So the pineal gland is the key player in the epithalamus, and the pineal gland releases melatonin. Most of us have heard about melatonin at this point. It helps to regulate that sleep-wake cycle, making us tired at night, right? There is another thing that happens in the epithalamus, and this is called optic. Let me try to spell it so you can read it. Optic chiasma. This is the crossing over of vision to create a 3D effect, right? So when you walk with both of your eyes open, you can see things three-dimensionally versus when one eye is closed and you don't have optic chiasma, you don't have a 3D effect. So again, epithalamus kind of has the main thing going on, which is controlling sleep pattern via the pineal gland and melatonin, and then also optic chiasma. And we also already wrote the function of the thalamus being the switching station for sensory information except the sense of smell. So again, this facilitates information going where it needs to go on those destination on that primary somatosensory cortex. The hypothalamus functions there is that it's going to produce hormones. So hypothalamus, you can think hormones, it produces hormones and it sends them to the pituitary gland to be stored. So pituitary gland stores and releases hormones, but it does not create the hormones. The hypothalamus produces the hormones, the pituitary gland stores the hormones. There is a tube or like a little channel that connects the hypothalamus and the pituitary gland. So they are connected by a tube. So I'll kind of circle the arrow to kind of point out that's what connects them. And that tube is called the infundibulum. Infundibulum connects the pituitary and the hypothalamus. Again, this is the majority of the hormones that regulate body function. So let's go majority, produces the majority of body hormones in the hypothalamus. So as you can see, diencephalon is mainly hormones, the epithalamus for melatonin and the pineal gland, the hypothalamus producing the majority of hormones for the body, storing it in the pituitary gland. The thalamus is kind of like a little third player, functioning to switch station, sending sensory information where it needs to go onto the right spot of the primary somatosensory cortex, so you know where that information is coming from. If you touch your lip, I know I'm touching my lip because the thalamus sends that information to the lip part of the primary somatosensory cortex. Good? All right, so let's do brain stem. Let's go for a different color. Let's go for green. The brain stem, the midbrain, doesn't really technically have a function. It's a little odd. It has the cerebral aqueduct. We already talked about the cerebral aqueduct. It connects the third to the fourth ventricle. So I'll just write third. Oh my, you might not be able to read that, it's so small. Third to fourth. Connects the third to the fourth ventricle. It also has the cerebellar peduncles. So cerebellar peduncles are little tracts that connect the brainstem to the cerebellum. So we're going to put, I'm going to put it over here because that's a cerebellum term, and then I'll tell you which ones. So peduncles. So hard sometimes to write straight. P. Tracts. This is not a D. It's a C and an L. Peduncles. Tracts that connect cerebellum to brainstem. So the midbrain has the superior cerebellar peduncle. The pons has the middle cerebellar peduncle. Again, this is just a tract, which is a myelinated group of axons that's connecting the cerebellum to the pons. Then the medulla oblongata has the inferior cerebellar peduncle. All right, now for the function of the pons. The pons functions as the respiratory center. That is its main function. It also has a little bit of a function for switching information, kind of like how we talked about with the thalamus. The medulla oblongata is the inferior cerebellar peduncle. And in it, we have the pyramids. The pyramids are for decussation, contralateralization, and chiasma, right? The exchange of that information. This is basically helping central white matter to cross over from the left to the right side. So the pyramids allow info to cross from left to right and vice versa. So remember when we talked about, like right here, right below it actually, where I said the cerebral hemispheres are separated by the longitudinal fissure. The corpus callosum helps connect the two halves. And then I talked about contralateralization, which happens 95% of the time. And then ipsilateral, which happens 5% of the time. Well, the pyramids in the medulla oblongata are what allows the two halves of the cerebrum to communicate with each other back and forth. In addition, the medulla is really important, and this is something you'll come back to when you're in nursing school as well. There's the cardiac center. The cardiac center, remember the center is a very, very large group of cell bodies. So the cardiac center controls heart rate. There's a vasomotor center. The vasomotor center constricts and dilates blood vessels. If you constrict and you dilate blood vessels, what do you control? You're controlling blood pressure, right? Making blood pressure go up or making it go down. And then finally, we have our medullary respiratory center. Medullary respiratory center. This is the last one. And this one's a little different. Let's do this so it's a little less confusing. Okay. So our function of this one. So the medullary respiratory center controls respiratory rate. And then it also controls coughing, sneezing. I know that doesn't really look like sneezing, but I think we can work with it. Swallowing, gagging, and vomiting. So the main respiratory center, I just don't want you guys to get confused. The main respiratory center for the test is in the pons. The medulla has the cardiac, vasomotor, and medullary respiratory center that helps with controlling respiratory rate and heart rate and blood pressure. Okay, but the main respiratory center would be the pons. This is the brainstem. Remember when we hear like of a patient in a hospital and we say, oh yeah, well, they had a brainstem injury. Usually you would know that that patient would be unlikely to survive off of a mechanical ventilator, right? Because it's a machine that's breathing for them. Okay, and usually we have to give them medications to control their heart rate and their blood pressure. Why? If you have a brain injury to your brainstem, you no longer have the ability to control your respiratory center because your pons was damaged. You can't control your heart rate because your medulla oblongata cardiac center was damaged. And you can't control your blood pressure because your vasomotor center was damaged. So this is why a brainstem injury is such a big deal, right? Because that's damaging those two sections that control your blood pressure, heart rate, and breathing, okay? People can recover from a brainstem injury. Miracles do happen. It's just that it's not a common thing. Cerebellum is our last part, right? And we'll see how much I can fit. There's not a whole lot to the cerebellum, but I don't know if I can fit all that there. We'll erase this peduncles part so I have some hope. So cerebellum is the second largest part. Second largest part. Our largest is obviously our cerebrum, right? Largest part of the brain is the cerebrum. Second largest is the cerebellum. It has three regions. It has a superficial region. The superficial region is the outer cortex. So remember how I told you that the cerebellum has gray and white matter? Well, here it is. The superficial region is the gray outer cortex. The deep region is kind of like those deep white matter. So this is the internal white matter. And these are called arbor vitae. Arbor. V-I-T-A-E. Vitae. Arbor vitae. And then lastly, we have our ultra deep. And the ultra deep are cerebellar nuclei. Remember when we said we had cerebral nuclei and they were ultra deep? This is cerebellar nuclei. So 1, 2, 3. Then it has folia, which are going to be its convolutions. So kind of like the cerebrum had convolutions. The folia are the convolutions on the cerebellum. The vermis. This is the septum where the two sides of the cerebellum meet. There are two cerebellums and they meet at the septum. So this is the meeting point of the two sides. All right, so that's the three regions, the folia and the vermis. And then I think I might be able to fit it up here. I can erase this. So remember, these are the 4 regions of the brain. Let's put this here. 4 regions. 1, 2, 3, 4. Okay, so what is the function of the cerebellum? So it has one function, proprioception. Proprioception is knowing the location of the limbs. So proprioception, knowing the location of the limbs. So when you lay in bed at night and nothing is touching and it's dark in the room, you know where your feet are because of the cerebellum, proprioception. It also functions for fine skeletal muscle and motor control. Right, so fine skeletal muscle, motor control. This looks like equilibrium and posture, giving you that sense of balance, fine motor movement. This is called, Newton always calls this the cat brain, because it's that, you know, when a cat balances on like the top of a little like fence, we would call that the cerebellum that's being activated. All right. So that's your outline of the brain chapter. It should be the majority of what you need to know. And then lastly, I want to talk about limbic system and cranial nerves. Okay. So let's see, where do I have room? So there was the brain. We talked about our CNS, PNS terms, meninges, gray versus white matter. Limbic system, like I had said, is for emotions. Limbic system is how you, I'm going to put it here because this is where we talked about it before. Limbic system is how you process and store emotion, right? So like if you watch a movie and it makes you cry, that's because of the limbic system. This includes smell. So remember how we kept saying smell, it doesn't include smell, it doesn't include smell. This does include smell, and we call this habenacular nuclei. Habenacular nuclei are our emotional response to odor. Emotional response to odor is the habenacular nuclei. So for example, you know, it could be positive or negative, right? You smell something totally disgusting and it makes you nauseous. That could be habenacular. Maybe you smelled bad food one time and it made you sick, and now when you smell it, that same food again, even if there's nothing wrong with it, it still makes you feel nauseous, right? Habenacular. Maybe you smell, you know, a certain like ingredient, like your grandma used to cook, you know, this certain type of chicken, and when you smell that herb, you feel those same feelings again of being with your grandma. That would also be habenacular. So it could be positive or negative, but it's an emotional response to odor. So this is to smell, right? So that's the limbic system. It's not too complicated. I already showed you the different nuclei of the limbic system, and we talked about what it is, right? It's more like a general thing in the brain, kind of located around the brainstem. All right, so overall, we talked about the brain, 4 regions. And then right here, it says cranial nerves. So I'm about to show you the cranial nerves and what you need to know for the test on that. But this is the outline of the brain. We talked more about CNS parts, PNS terms, and meninges. We talked about gray versus white matter and nuclei. And then we talked about the 5 lobes specifically because we couldn't get all that to fit on the main outline. So now, cranial nerves. Then I'm going to show you, I'm going to write down what you need to know, and then I'll kind of show you the video that I used in the module. So for the test, you need to know the Roman numeral. So 1, 2, 3, 4. So V represents 5. X represents 10. So when we take a V and we put a 1 behind it, it would be 5 minus 1, 4. When we add a 1 in front of it, it's 5 plus 1, 6. And then one of the things that they do with Roman numerals is they draw a line on the top and the bottom. It doesn't change anything. That's what I did over here. So this is 5, 6, 7, 8, because we keep adding 1 more, right? 5, 6, 7, 8, 5, 6, 7, 5, 6. Does that make sense? So you do need to know the Roman numeral for the test, not just the number. IX would be 9. We take 10 and we subtract 1 by putting a 1 behind the 10. Then there's 10, and then X1, add 1 to the X, so that's 11. And we can also draw the line with these, and then XII, 12. So you do again need to know the Roman numerals, 1 through 12. Then you need to know the names. So you need to memorize the cranial nerves 1 through 12 and their names. So 1 is olfactory, 2 is optic, 3 is oculomotor, 4 is trochlear, 5 is trigeminal, 6 is abducens, 7 is facial, 8 is vestibulocochlear, also called acoustic, 9 is glossopharyngeal, 10 is vagus, 11 is accessory, and 12 is hypoglossal. And then you need to know their basic functions. So I'm going to write for you what would be the most important things to know for them. Olfactory is for smell. Optic is for vision. Ocular motor moves the eyes and prevents eyelid drooping. Trochlear moves the superior orbit of the eye. Trigeminal performs mastication and facial sensation. Abducens performs eye abduction and prevents double vision. Facial is for facial expression. And 2/3 taste sensation. Vestibulocochlear or acoustic is for sound and body balance. Glossopharyngeal senses taste from the 1/3 back of the mouth. And for swallowing. The vagus is for speech. The accessory is for shrugging the shoulders and head resistance. And then the hypoglossal is for tongue movement. It may feel a little overwhelming. I do remember multiple questions on my exam on this. A lot of them were on the Roman numeral and the name, and then I also had others on the function. I will tell you that for those functions that I just listed on this page here that you see, that is sufficient for the exam. There's a table in your book that I think you're referred to that has a lot more information, and you don't need all of that information for the exam. You can just use, again, what I put right here, okay? So you don't need all of that. You can just screenshot what I have here, and this should be sufficient, okay? All right. We only have 15 minutes. And that's not enough to do the brain model because there's quite a bit on it. So I think what I'm going to do is I'm just going to kind of end this session and leave the last 15 minutes for questions. If you guys have questions. And then on Thursday morning, we'll start with probably, I don't know, we'll see what you guys want to do, but we'll do some torso models. We'll do the spinal cord lecture. And then we'll go back over to like brain and spinal cord models is probably what we'll do, okay? So I will see you guys Thursday morning. but please don't leave unless you guys have questions. If you guys have questions, eye abduction would be like looking to the side, right? Because abducting something is bringing it away from midline. So if I'm looking over in the corner, I'm abducting. Does that make sense? Adduction would be bringing it back to the middle. And if you guys need to go back to any of the screens as well, we can do that. Thank you.
Brooke, I have a question.
Go ahead.
Um, are you going to do the SI Thursday, not Friday?
Yes, it's going to be Thursdays from now on,
Oh,
because I used to have it Fridays during March, but my clinical days changed in nursing school. So now it's going to be on Thursdays.
I see.
But I believe the sessions are recorded and I try to update those. Those should be available to you in the module.
Oh, because I was like wondering, I like Friday because I'm working Thursday and going to school too. Thank you though.
Yeah, well, the recording should be on Canvas, so you could watch them on Fridays.
All right.
Yeah.
Thank you.
Yeah, let me know if you need any help finding them at all.
Yep, sure.
Okay. See you soon.
See you.
Thank you.